Good morning, students. Today's topic is cell line banking and cryopreservation. A cell bank is a facility that stores cells of specific genome in a frozen viable state so that it can be used for medical use, research, and commercial product manufacturing. Each ampule in the cell bank is derived from a single pool of cells. The cell bank system consists of two tiers, the master cell bank and the working cell bank. The master cell bank is a collection of uniform composition of cells derived from single tissue or cells which are cryopreserved. The master cell bank are derived from immortal cells or in some cases primary cell cultures. The master cell bank for diploid cell lines should be prepared from cells at low population doubling level. The working cell bank is derived from the master cell bank. This is a generally accepted approach to provide a continuous supply of cell substrate for research work, medical use, and for manufacture of cell-based products in industry. Steps for generating a cell bank. The first step is the selection of source cell for cell bank. It could be from primary culture, an immortal cell line, or a stem cell. A stable cell line is developed. Second, the selected cells are cultured and passaged. At each passage, the growth kinetics is monitored, the activity of the cells are verified by bioassay, the cells are multiplied under aseptic conditions. All relevant details as to source, passage, cell doubling, bioassay are recorded. Next, freeze 2 to 5 million cells per vial to produce a master stock. A minimum of 10 vials are made. The vials are checked for contaminations like mycoplasma, bacteria, etc. If the tests are negative, one of the master stock cell is thawed to create working cell bank. Around 20 plus working cell bank vials are made. The working cell bank vial are freezed. Then randomly one of these vials are taken and subjected to bioassay, characterization and other quality assays. Records are maintained for all the validation assays. The master cell bank needs to be monitored and well documented. The morphology of the cells, the species of origin, the growth kinetics, split ratio, doubling number, bioassays, contamination tests are done and recorded. All quality and validation checks need to be conducted. If the master cell bank has cells that have certain specific characteristics, data to demonstrate it should also be recorded and validated. Example, if a cell contains an expression gene to produce recombinant protein, data should be obtained and demonstrated to show the copy number and physical state of expression and the quality and quantity of the protein produced. The working cell bank should also be validated for quality checks and recorded. The master cell bank and the working cell bank are generally kept at different places. This is to avoid loss of cell substrate due to a local disaster or equipment malfunction. Access to these cells are limited and the inventory of the individual ampules are well recorded. The diagram below illustrates the flowchart for documentation of cell bank and validation. Next, cell cryopreservation. The principle of cryopreservation of animal cell is similar to that of plant cells. The steps followed are cell selection and development of cell line, cell passage, harvesting of cells, addition of cryoprotectants and pre-treatments if required, freezing, storage, thawing, recovery and reculture, measurement of survival and viability. Cell selection. The cells from the source are passaged and maintained in aseptic conditions. The cells are validated and quality checks are done at regular intervals. The cells are examined for all signs of microbial contaminations. It is facilitated by growing the cells in antibiotic-free media for several passages prior to testing. This allows time for any hidden resistant contaminants to reach its higher level and can be detected. The cells at the end of the log phase of growth are selected for cryopreservation. Prior to harvesting the cells, the nutrient media is changed. After all the quality checks and validations are done, the cells are then harvested. The harvested cells are counted and diluted. The whole procedure is carried out in aseptic conditions. The cells are kept in chilled environment to slow down the metabolism and prevent clumping of cells. Gassing with carbon dioxide is done to maintain the culture pH. Next, cryoprotection. Cryoprotectants are added to the cell. Both permeable and non-permeable cryoprotectants can be used depending upon the cell type. DMSO and glycerol are the most common cryoprotectants used. While using cryoprotectants, the toxic level of the cryoprotectants should also be checked. Hence, these should be used in lower concentrations. Example, for DMSO, the final concentration is 5 to 15% volume by volume. In case of DMSO, if one-time addition of DMSO is found to be toxic, it is added in multi-steps. After the cryoprotective solution is mixed with the cell suspension, the resultant cells is added into small storage vessels in aliquots. Plastic vials are replaced glass ampoules for cryogenic storage due to their greater safety and convenience. Care should be taken that there is no leakage as well as pinholes in these vials. Next, labeling and record keeping. All the vials are labeled and the records are kept. The records include the source of the culture, the identity, the date of frozen, the cryoprotectants used, the passage number, the split number, population doubling levels, quality control tests done, etc. Details of this has already been discussed in the earlier session. Slow freezing and vitrification are used as the common cryopreservation method for most of the animal cells. Both these methods have been discussed in detail earlier, stating its advantages and differences. In case of slow freezing, cooling rate of -1 to -3 degrees centigrade is found to be satisfactory for most of the cell cultures. However, it needs to be standardized based on cell type. Two different types of freezing method is used. Controlled rate cooling using programmable freezers or passive cooling. Passive freezing involves placing the cells in freezer about -80 degrees centigrade or -150 degrees centigrade and allowing the samples to cool in an uncontrolled fashion. From the cooling chambers, the vials are then transferred into liquid or vapor nitrogen freezers depending upon the temperature requirement for storage. For recovery of cells, the first step after cryostorage is thawing. Rapid thawing provides the best recovery for most of the cells. The vials are taken from storage and placed in warm water and agitated gently. The recovery of the cells are then done. The cells in the vials are transferred into suitable media and incubated. After a majority of cells have been found to be attached to the media, the cryoprotectant media is removed and replaced with fresh media. For the cells that are sensitive to cryoprotective agents, the media containing the cryoprotectants are removed by centrifugation. The cell pellets are then resuspended in fresh media and incubated. When glycerol is used as a cryoprotectant, the addition of large amount of fresh media may lead to osmotic shock for the cells and destroying the cells. In such case, stepwise dilution with an equal volume of warm media every 10 minutes is done. After the recovery, the viability of the cells are checked by using different dyes and the viability percentage is calculated. The figure here outlines the different steps taken for cryopreservation of cells and certain freezing checklist. With time, the conventional method of cryopreservation of cells is modified for easy handling of the cells. Example, on-chip direct freezing and thawing. In this method, the cell suspension, which contains the cells along with the cryoprotectant DMSO and fetal bovine serum and nutrient medium are injected into chips. These chips are then inserted into centrifuge tubes filled with isopropyl alcohol. These centrifuge tubes are then maintained at -80 degrees in cryogenic refrigerators. Thank you.